Our next speaker is going to be Dr. Marina Pajic, who works in the pancreatic cancer group and is going to be talking about the work she's been doing there on sequencing cancer genomes.
Okay. All right. Hi, everyone. Well, thank you for that introduction, and thanks a lot to the organizers for allowing me to talk to you today and to show you some of our recent results that we're hoping to publish on developing personalized medicine strategies for patients with pancreatic cancer. So pancreatic cancer is a devastating disease, very poor overall survival rates of only 5% of patients being alive 5 years post-diagnosis. And as you can see here, there are very few treatment options for these patients. Surgery is currently the only potential for cure for a very small proportion of patients who qualify, and so that raises this survival from about 5% to 15%. So there's clearly a lot more that needs to be done about this. Now, both our sequencing initiative as well as those of many others have shown now that cancer is a heterogeneous disease. And specifically for pancreatic cancer, most aberrations appear at a very low frequency of only about 2% or less. So this could potentially explain the failure of current clinical trials with novel agents where your responsive phenotype is just too low to detect. And so that's just illustrated here in this pictorial here for pancreatic cancer. So our current challenge now is really how to identify and better prioritize some of these targetable phenotypes for the disease. And so our group, together with Sean Grimmon's group at University of Queensland and many other collaborators across Australia, formed the Australian Pancreatic Cancer Genome Initiative, or the APGI. And so our aims as part of the large International Cancer Genome Consortium are to sequence a large number of pancreatic cancer samples. So these, our aim is to get to 400 pancreatic ductal adenocarcinomas. And here you can just see the patient journey going from top to bottom. You can see what happens to the patient as he or she enters the APGI study. So we're looking at primary operable, so patients with primary operable disease here. On the x-axis, you can see what we call the sample journey or the genome journey. So what happens to the sample as it leaves the patient. We have extensive quality control, analyte prep. The samples get sent off to Sean's group for sequencing. And what we really would like to do here is, where possible, to directly use the genomic sequence data to guide the clinical management for these patients. But for some of those more novel mutations, we have also developed preclinical models in the lab to functionally validate some of these. And so just to show you what sort of models we're working with, so these are the patient-derived xenografts and patient-derived cell lines. So again, you have a pictorial here showing you the tumor resection and the sample heading off for profiling. In parallel, though, we also get a tumor piece that we then engraft into immunocompromised mice. After this engraftment stage and several rounds of expansion to generate a significant amount of tissue, we establish what we call a patient-derived xenograft cohort. And so currently we have about 90 individual patient-derived xenografts that we then use to generate patient-derived cell lines. We have a panel of 20. We use these cell lines for various functional screens that we've got going both at the Garvan at the moment as well as with some international collaborators. These so cell lines have been sequenced now. The xenografts we're also looking at putting into the pipeline for sequencing as part of the ICGC. And in parallel, in the lab, we're also running what we like to call the personalized medicine preclinical trials using these xenografts. So really trying to test newer therapeutic strategies and targeting these based on the individual biology of each tumor. And one nice feature of these xenografts is that histologically they do resemble the primary tumors that they come from. So they still incorporate a considerable amount of stroma, and stroma is an important characteristic of pancreatic cancer. Genomically, and this is just showing you some SNP chip data. So here we have the log-R ratio on the y-axis, and on the x, so just going from left to right, you've got the chromosomes going down the line. we've got the patient tumor DNA on the top and the xenograft DNA on the bottom. So this is actually enriched epithelium of the xenograft DNA. And what you can see here is that these deviations from zero, so in the patient sample, indicate aberrations, so deletions, gains, and they're nicely mirrored in the xenograft. So what are some of the potentially targetable molecular phenotypes that we have identified? So actually at the moment we've got about 19 that we're looking at. So this is both looking at published data in other cancers as well as some of our analyses. But here I'm just focusing on some of the more directly targetable or druggable phenotypes. We've looked at so to identify these, we've used evidence from clinical studies. So for example, we know that there are case reports of patients who have been given mitomycin C and have had dramatic responses to this. We are looking at emerging phenotypes in other cancers, so HER2 amplification. It's being targeted in breast cancer, so with trastuzumab, so we're also looking at repurposing some of these. And also there is a range of novel phenotypes that we're also testing in our preclinical models. So just the focus today will really be on some of the clinically approved agents and how we're trying to develop personalized approaches based around them. So the strategy we're using, we're looking at the first 88 APGI patients that have been exome sequenced and have had gene expression profiling done. Of those, 47 have also been whole genome sequenced. And we're using the NCI phenotype to genotype and genotype to phenotype strategy to match these phenotypes. What this really just means is that we're using, so we're measuring clinical responses and then analyzing these, performing genomic analyses on these patients to identify potential molecular phenotypes. And at the same time, for more known aberrations, we directly use a genotype to phenotype approach. So we try to guide palliative therapy for some of these patients. So at the moment, of the 88 patients in this study, and you just have to realize that this is a prospective cohort. It's a young cohort, so the data is still not completely mature. So of these 88, 61 patients have recurred so far, and 27 have received some form of palliative therapy. The reason why we're looking at only palliative responses is because here we can really have an objective measure of response. So we use RECIST criteria, and we've got measurements of CT scans, so you can objectively measure how this patient responded, as well as measuring serum marker levels. And specifically the one I'll be talking about today is the CA99 serum marker. And we have also screened a range of patient-derived xenografts as well based on the predicted signatures. So just to very quickly run through, so these are the types of agents that the patients would have seen, the 27 patients, and how we're stratifying them according to different phenotypes. So these are some of the phenotypes that we're looking at. So we've got gemcitabine here, and we're looking at outliers of expression. So this is using gene expression profiling in specific nucleoside transporter proteins. Subsequently, we've got platinum and mitomycin C response and looking at high genomic instability as well as mutations in the known DNA damage repair genes of interest that could predispose to sensitivity to these agents, and some of the other more rarer phenotypes. So what I've told you previously is that each individual phenotype is actually on its own quite rare. So the power in this, developing the concept of personalized medicine for pancreatic cancer really has to, we really have to look at the combined effect of the on-phenotype therapy. And so this is what we've done here. So 10 patients of the 27 who have received palliative chemotherapy have received some form of on-phenotype treatment. 17 were in the unselected or off or unknown phenotype group. And although this is a small study so far, what it becomes very clear is that there is a dramatic increase in the median survival of patients who were on phenotype therapy. And 6 of these were exceptional responders, and I'll just very briefly mention a few of those. So markers of gemcitabine responsiveness. So we really went just into the literature. And so there are 3 human nucleoside transporter proteins There's actually several others, but these are the three that have been implicated in some way in preclinical studies to relate to gemcitabine response in cancer cells. So we've looked at the expression of these three in our microarray APGI cohort. And so this is what the data looks like. So here we have just rank expression of the three genes, relative expression levels, and so each little dot represents a patient. If we just look at the top expressors, so we've got 13 patients here, and we subsequently looked at the response of these patients to therapy. What becomes really quite clear is that 3 responses, so 3 patients received gemcitabine in this cohort, and all 3 had a measurable response to gemcitabine. One was an exceptional responder, and I'll just very briefly talk about that. Because this is one case where we were actually able to guide palliative therapy. There were 2 partial responses in the off or unknown phenotype group, so 7 patients received gemcitabine and none of them responded. And so this particular patient, actually at the time of recurrence, we were looking at this data. This was the top, the second highest expressor for HENT1, so marked here in red. And we've also had a range of xenografts profiled, one of which was this patient xenograft, and I'll show you that on the next slide. So based on our data, the oncologist was happy to put this patient back on gemcitabine, and you can see the dramatic drop. So this is just CA99 levels, so this is a seromarker of treatment response. You can see that at the time of recurrence, the levels start to rise dramatically. Following palliative gemcitabine, the levels drop back down. And so this patient did quite well for a significant number of months still. What do the xenografts look like? So this is the specific xenograft that I was telling you about. So here we have 5 xenografts out of the 13 patients that were outliers. So we had 5 available to us for screening. So we've screened all of them. And just showing you tumor volume on the y-axis here. You can see the mass, the tumor responses to gemcitabine in blue compared to some of the other DNA damaging agents and saline control in the black. Upon each relapse, we've also retreated our tumors with gemcitabine because we're interested in developing models of acquired resistance, and we also wanted to see how reproducible and how robust this response really is. And so these 5 tumors remain repeatedly sensitive, so we've got 5 out of 5 on-phenotype responses, and in the off or unknown phenotype group, none of the xenografts responded to gemcitabine. And so these 2, unfortunately, were, so these are the 3 patients that responded in the clinic that I've shown you in the previous slide. These 2 were, unfortunately, the patients had surgical deaths, so they did not ever see any gemcitabine. Now I'll just very briefly go over some of the analysis that was actually done at University of Queensland, so by Sean Grimmond and Nick Waddell. So they have performed whole genome sequencing on 47 thus far patients in the APGI cohort. And based on analysis of the numbers of different types of intrachromosomal rearrangements, deletions, gains, and point mutations, they were able to robustly classify pancreatic tumors into 4 different classes. So we have a class of tumors which are unstable or have pangenomic or high genomic instability. Each of these each of these lines here basically shows you a single aberration. So you can see that this tumor is just hugely, hugely rearranged. We have tumors with more focal gains or losses, tumors that are quite stable, and tumors with potential whole chromosome loss regions. So we predicted, based on some of the data in other cancers, again, breast cancer is probably one of the good examples, is where high genomic instability predisposes to sensitivity to certain DNA intercalating agents. So we have screened 4 xenografts so far, and I'm showing you 3 here. One was a tumor with a known hereditary mutation in one of the DNA repair genes, BRCA2. Cellularity was just, so tumor cellularity was just too low, and this is a frequent problem with pancreatic cancer, so not sequenced yet. 2 xenografts with high genomic instability, and now focusing on the green and red responses, all tumors repeatedly sensitive to DNA damaging agents. and not particularly sensitive to gemcitabine, especially these two. Just to show you an example of what a stable genotype tumor looks like, basically no response. Finally, we were also able to, what did I just do? Oh, okay, it's fine. So finally, we were just able to, based on this signature, we're able to guide palliative therapy for one patient so far. And so this is, again, showing you CA99 levels here. The patient has recurred, CA99 levels increasing dramatically. The patient was put back on Folfox in this case. So this is oxaliplatin, an analog of platinum-based chemotherapy. And the patient is still alive 40 months post-diagnosis. Again, showing you on the CT scan, you can see this lesion here appearing at the time of recurrence, and there is complete resolution of the lesion. So in this, for this specific phenotype, we've got on-phenotype responses, 4 out of 4, and 3 exceptional responders. 1 was a return of information to patient, and in the off or unknown phenotype response group, 1 out of 5 in the clinic. So what I've hopefully shown you here today is that there is some hope and potential in treating pancreatic cancer based on this, based on molecular subtype. It would hopefully lead to avoiding inappropriate toxicity and giving inappropriate therapy. We're also using this to now test much more novel agents which haven't seen the clinic yet. And finally, just a little add that we are doing this now in a prospective manner in the impact, using the impact personalized medicine clinical trial for pancreatic cancer, which is a phase 2 study now open. And so this is not just me doing this work, so there is a whole bunch of people all around, both in our group, Queensland and Australia. And also I would just very quickly like to thank the patients, because without them we wouldn't be here. Thank you.
Okay, we've run a little over time. Is anyone who has a quick burning question? All right, I'll just quickly ask a question, because even though I'm running over time. I was just wondering, have you been able to see any acquired drug resistance and then being able to resequence a tumour and see the reason for that?
That's what we're doing. So actually not with the gemcitabine response, but with the DNA intercalating agents. So we're looking at PARP inhibitor therapy. We've got resistant models. They're being sent off for sequencing. We anticipate that it will be reversion in the BRCA2, initial BRCA2 mutation that's present in here, but we've got no evidence of that. We're working on.
All right, thank you. Okay, our next speaker is Dr. Marina Pajic, who did her PhD in the US and has now come to work as a postdoc in David James's lab.